for DNA. How did we fit a very large molecule into the small nucleus? How did we do that? Okay, how? All right. So we're gonna condense the old DNA by wrapping it around, right? Histones. Okay. And histones basically we have five histones, H1 to H5. H1 is a linker histone. It links uh, DNA, but the ones that form the core of the histone around which we wrap around DNA, right? Those are going to be H2 to H5. Yeah. So, what is the major amino acid component of histones? What what kind of amino acid are they made up of the most? Mhm. So arginine, right? That's one of the major components, arginine. So basically, the basic amino acids, those amino acids that can form positive charges, right? They are the major components of histone, and we're gonna see how that is relevant. And arginine is kind of like the most abundant, right, amino acid in histones, and that is why arginine is seen as an essential amino acid, right, in children, because during growth, where cells are actively replicating and dividing, they need to also make copies of histones, right, as they are making DNA. So there's a high demand for arginine in any situation, right, that leads to increased growth, increased cell growth, increased cell proliferation, right, or division. Arginine will be an important amino acid, right, in those scenarios. Yeah. So in children, in people with cancer, they're going to be at higher risk for arginine deficiency because cancer cells are going to be using, right, all the arginines to make histones, to proliferate, to divide. Yeah. In patient that is very sick and healing, maybe after severe burn, yeah, burn wound, severe trauma, right, where the body is healing, they're going to require a lot of arginine. Yeah, because cells will be proliferating, right, for that healing process. All right. So it makes arginine essential in those scenarios. So how do we? Okay, so we know we wrap DNA around histones and all that, but what other component plays a role in packaging DNA? So we know what we use to package it. We need histones to do that. But what other property of histones and DNA make it possible for you to tightly band them together? Okay, when you bind them together, you form the chromatin, right? So chromatin is basically DNA wrapped around histones, right, and some other supporting proteins to form that condensed form, right, in the nucleus. But remember also, what also plays a role with what charges, right? Yeah. So what is the overall charge of DNA molecule? It's negatively charged, right? Remember, DNA contains phosphate backbone. So the phosphate backbone of DNA gives DNA an overall negative charge. And we just mentioned that histones basically contain what? Mostly basic, right? Amino acids. Which amino acids that give histone, right? An overall positive, right? Charge. What do you what do unlike charges do? Unlike charges, what would they do? Do they repel each other or attract each other? Attract. So unlike charges attract. So that also contributes to what? Packaging DNA. Having an histone or having histones rather that are positively charged and DNA is negatively charged, right? Besides wrapping them around each other, the charge also stabilize right, condensation, because they're going to be attracted to each other. It's going to lead to a tightly bound, right, or tightly wound DNA molecule. All right. So if I want to make DNA more tightly bound, like I want to make the relationship stronger, what can I do to DNA and histones? I want to make that condensation tighter. What can you do to DNA and histone? How can you modify their structure? You add more to tightly bound, right? Yeah. If you want to form heterochromatin, what will you do? Or what can you do? You're gonna you're going to acetylate, because acetyl is what charge? Negative. So if I acetylate DNA, acetylate histone, what happens? I'm adding more negative charge, I'm adding more unlike charges. Right, and they are going to what? Be tightly bound. There'll be more bond formed between DNA and histone. Sorry, should be positively charged. Yeah, positive, positive. Methylation, right? Methylation, no acetylation. Bound to tightly bound, right? Methylation. M for methylation, M for mute. Yeah, methylation, yes, methylation. So methyl groups will be positive charges, right? So if I had positive charge to histone, right? I'm adding positive charges to histone. I may add some positive charge also to DNA, but DNA already has a negative charge anyway. That is not going anywhere. So basically, you're adding more unlike charges, charges that is different from what DNA is. There will be more attraction between DNA and histone. Yeah. So methylation makes DNA more tightly bound, right? And that whole structure we call it what? Heterochromatin, right? A tightly bound DNA. If DNA is tightly bound to histone, then it is not accessible, right? By enzymes. It's not accessible. So processes like replication, right, can take place. Transcription of genes can take place. So basically the DNA and the genes are what turned off by methylation. So M for methylation, M for mute, heterochromatin. And the opposite would be to hard word. If I want to make DNA loosely bound, hard word, a negative charge. Add more negative charges. Right now we're adding like charges. They're going to repel, right? Each other. Yeah. So acetylation activates genes. It forms euchromatin. Because its DNA is loosely bound, it's going to be more accessible, right, to enzymes. That concept explains a lot of things in the body. One of the things it explains is, even though every cell in the body have the full complementary, right, of genes, every cell contains every gene, all of them. Why do some cells express only some genes, right, and not others? So it explains that because those cells can preferentially, right, turn off certain genes and turn on other genes. So some genes will be in the form of what? Euchromatin. They're going to be acetylated and active. Some genes will be what? In the form of heterochromatin, right? They're going to be methylated and mute. And that explains how cells differentiate. It explains why we have different cells, why neurons are neurons, why muscles are muscles, why blood cells are blood cells, why skin cells are skin cells. Because neurons will express the genes that will allow them, right, become neurons, make myelin, make sodium channels, etc. for differentiation, right? make change into axons, etc. Make synapse. While muscles will become what? They're going to express genes that will make them muscles. Actin, myosin, right? Troponin, etc. And turn off genes, right? That will make them neurons. Why neurons will turn off genes that would have turned them into what? Muscles or any other cell. So differentiation. Yeah. The concept also explains why cells expressing the same protein, right? Same type of protein, no, why cells express slightly different types rather of the same protein. We also explain why cells express slightly different types of the same protein. For instance, how many types of creatine kinase do we have? Creatine kinase do we have? Nothing less than three subtypes. There is the slightly different type expressed by what? Skeletal muscles. There's a slightly different type expressed by the heart, CKMB, and there's a slightly different type expressed by what? Neurons, CKBB. Why? Because they can turn off or turn on, right? Some maybe the muscles will turn on the CKMM gene and turn off CKMB and CKBB, right? So that may also contribute to why same proteins will be slightly different, right? In different cells. Yeah. This concept also applies to even normal physiologic processes like um, just give my lionization, where one X chromosome, right, is turned off and only one is expressed, right, if there are two, typically in females. So the X chromosome that is turned off is basically turned off by what? By methylation and forms what? Bad bodies. And even in patients with disorders like Klinefelter or any disorder that leads to more than one X chromosome in the cells, right? We're going to see the other extra chromosomes will be turned off by methylation, and they're going to have bad bodies depending on how many extra, right? X chromosome the patient has. Yeah, still by methylation. Also, the concept of imprinting, which is a normal process where cells can preferentially turn off one copy of a gene instead of expressing both copies. So we know we have two copies of every gene, right? One from the father, one from the mother. Yeah. Some cells express both copies of a gene. Sometimes they're going to turn off one copy of a gene and express only one copy, either maternal copy or paternal copy. They do that by methylation. They're going to turn off, we call that imprinting, but it's a normal process. Turn off one copy of a gene by methylating that gene and express only one copy. The problem with that is if the expressed copy become mutated, either deletion or point mutation, right? Then they have zero, right? A functional gene, and that will lead to diseases like Prader-Willi, right? And Angelman syndrome. Yeah. Or if cells inherit two copies of a gene from the same parent, instead of one from each parent, inherit two copies of a gene from the same parent, what we call uniparental, right? Disomy, right? And if that cell normally imprints the copies of those genes, right, from that parent, then that's another way a cell may not have, right, any functional, right, gene. Yeah, that can also be a mechanism of disorders like Prader-Willi or Angelman syndrome. Yeah, you said right. Both copies will be imprinted. Yes, and they will be turned off. Yeah. Mhm. So those are just some examples, right, of how that concept can be tested. Yeah. So a gene is hue chromatin, which means it's accessible. The process of what? Making a copy, right, of that gene or making a copy of the DNA, we call that replication, right? Replication. Replicate something, make a copy of. And replication is important because